So chapter 12 focuses on what's known as the chromosomal basis of inheritance. And Mendel, when he did his experiments, remember he's the one that kind of discovered these hereditary factors, which we now call genes. And we now know that genes are located on chromosomes. And our technology has advanced to such a state that we can actually identify the location of particular genes on the chromosome. So you can see here, they've highlighted an area of the chromosome that isolates the gene they're looking at. After Mendel, we also discovered how to identify and describe the stages of mitosis and meiosis. And this led us to what's known as the chromosome theory of inheritance, which states that Mendelian genes have specific loci or positions on the chromosome. So loci might look familiar because it's the plural of locus. We know they have specific positions on the chromosomes, and then all chromosomes undergo segregation and independent assortment, which are laws you probably remember Mendel discovered with his pea plant experiments, and this happens during meiosis due to the behavior of the chromosomes. So we're now going to move on to another scientist whose name was Morgan, and he showed that Mendelian inheritance has its physical basis in the behavior of the chromosomes. So Thomas Hunt Morgan was a scientist in the early 1900s, and this section kind of shows you an experiment that he did to answer some questions he had about inheritance and how he used the scientific method to design an experiment to understand his results. So first, he wanted to select an organism to observe. So like Mendel selected the pea plants, Morgan selected a fruit fly known as Drosophila melanogaster, which you'll see referred to as Drosophila for most of the rest of the textbook. He picked these fruit flies for several reasons. Number one, because they produce a lot of offspring from one mating alone. And then in addition to that, you can breed a new generation every 2 weeks with even more offspring. And then most importantly, they only have 4 pairs of chromosomes that are easy to see with a light microscope. They have 3 autosomes and 1 sex chromosome. So he was able to observe the chromosomes and really study the ones that he could see because there was only 4. In doing this, he identified one type of the fruit fly as a wild type, and this is known as the normal phenotype fly because they are the types that would be found in the wild most commonly. These normal phenotypes in the future are always going to be referred to as the wild type. That's just the phenotype for the character that's most commonly observed in nature. So for the flies, the wild type fruit fly had red eyes. The alternative traits that are alternative to the wild type, whether it be one or several that are not the regular wild type, are called mutant phenotypes. And so in the case of the fruit fly, since the wild type had red eyes, the mutant phenotype he observed had white eyes. So you can see this is the wild type fly, and this is the mutant. In one of his experiments, Morgan mated male flies with white eyes, the mutant types, with female flies with red eyes, the wild types. And he did this similar to how Mendel did, where he put purebred with purebred. And what he observed, similar to Mendel, is that the F1 generation all had red eyes, which is what would be expected. However, when he bred the hybrids, what he observed is that the F2 generation only showed the classical 3-to-1 red to white ratio, but only males had the white eyes. And so in observing this time and time again, Morgan concluded that there must be some correlation between the eye color and the sex of the fly. In further experiments, Morgan was able to determine that the white eye mutant allele must be located on the X chromosome, and this finding really supported the idea that genes are found on chromosomes, and we'll talk more about how he discovered this in a little bit. But this is just an example of what his experimental results looked like. So we had a wild type female with red eyes crossed with a mutant type male with white eyes. And in the F1, like we said, all the offspring had red eyes. With our results when we crossed these F1 hybrids, we observed the 3-to-1 phenotypic ratio, so most likely this would be a homozygous dominant, these would be heterozygous, and this would be the homozygous recessive. However, in examining the gender of the flies, remember, they noticed that only the males really ended up with the white eyes. So this demonstrates to us that the genes must be located on the chromosomes, because we know that the males have an X and a Y and the females have an X and an X. So if the wild type gene is located on the X chromosome, then the female is always going to pass that gene on. And the male is going to pass that on if he has a daughter, because he has to pass on his X chromosome. However, if the male doesn't get the wild type gene, he has a recessive perhaps, and he doesn't get another one on the Y chromosome, then he's going to end up having white eyes. So we will come back to explain this more deeply in the next video.